It's a great pleasure to be here among esteemed colleagues, and a great honor to be here for celebrating Bob's amazing career. So what I'm going to do, I will provide an overview of the work we have done over the past 15 years, and I will share with you how our thinking has evolved around the role of sweet taste receptors outside of the tongue. So all classes of nutrients, macronutrients, primarily fatty acids, amino acids, and sugars can enter cells and can have different fates. Some, they serve as building blocks, for anabolic reactions. Some others are more suitable for energy production. But what is probably relevant to today's discussion is through the metabolites, they can engage with intracellular sensors to govern signaling pathways that they maintain cell homeostasis. But of course, I don't need to emphasize in this audience that over the past 15, 20 years, it has become apparent that the same classes of macronutrients can also be independently sensed by cell surface receptors, G-protein coupled receptors, that engage to acute signaling events also to govern cell homeostasis. So in my eyes, there are two modes of nutrient sensing. One is the classical one that involves nutrient uptake and metabolism, and the other one is receptor-mediated signaling. And my lab over the past 15 years has focused on sweet taste receptors, which, of course, is formed as a heterodimer between two proteins, T1R2 and T1R3. And this is a slide that summarizes approximately what we have done. As you can see, all my work is outside of the tongue. And the idea here is, what is the role of these GPCRs? What are the ligands? And then for many of these organs, we had to first pioneer the physiology, since there were no literature, and then ask whether they play a specific role in disease. But why am I here and how everything started? So in 2008, I joined Sanford Burnham Prebys to study islet biology through work of Bjorn Tirbert. And 3 months in, Tim Osborne was recruited and eventually became the director of the major metabolism program there. And during his recruitment seminar, he presented his recent work back then, suggesting a role of bitter taste receptors in the gut. Of course, back then for me it was all unknown, but I remember how excited Bjorn and I were that there is a potential for these receptors to be in beta cells. But I was a little bit skeptical of what would be a role of beta receptors in beta cells, where probably you wouldn't expect noxious compounds to circulate into the bloodstream. And then when I start digging through the literature, inevitably I became immediately aware of the recent work with two seminal papers by Bob, establishing the role of these receptors in enteroendocrine secretion, and gave me hope that through sweet taste receptors, which seems to me to be the more logical choice, at least back then. So the goal back then was to understand how an intestinal pancreatic axis operates through these receptors in the regulation of homeostasis. And we first showed that all the machinery is there in human and mouse beta cells. And we developed a reporter mouse, and we confirmed that indeed T1R2, as you can see here, is expressed specifically in beta cells, but not in alpha cells that they are responsible for the secretion of glucagon. And what we did back then is we gave a small amount of fructose in addition to glucose through an intraperitoneal injection. And as you can see in wild-type mice, as expected, the black line, glucose elevated the levels of insulin. This has been demonstrated, of course, before, but addition of fructose potentiated these effects, and these effects were absent in T1R2 knockout mice. We also show the relevance directly to isolated islets, and as you can see here, addition of 3 millimolar fructose, which is approximately the level that one finds in the circulation after a mixed carbohydrate meal, potentiates the effects of glucose, but this is only in wild-type islets, not in islets that are lacking T1 or 2. And thankfully, the system is preserved in human islets. Again, you can see fructose here, but when we blocked taste receptors, not T1 or 2, but T1 or 3 in this case, with lactosol, we abrogated the potentiating effects of fructose. In terms of the signaling, actually, the signaling is very well preserved in beta cells, similar to the taste cell. Through a number of biochemical studies, we show the involvement of PLC, calcium flux, and of course the dependency of tryptophan dependent depolarization. We use, of course, tryptophan knockout mice from Bob, and through this work we show that indeed, to sweet taste receptors, they potentiate insulin secretion by further depolarizing the cell. And this is not only confined to fructose, but all other, artificial sweeteners like saccharin here. This is through turf microscopy that directly assesses events at the cell membrane. And you can see upon stimulation of saccharin, you have an immediate activation of PLC, shown in the black line, which precedes the rise in calcium, further suggesting, a role of this signaling mechanism. And of course, when we inhibited pharmacologically PLC, we didn't affect the effects of glucose, but we affect the potentiating effects of saccharin. Okay, so back then when we were working, it became immediately apparent that the route by which we administer glucose in a mouse alters the effects that we see. And what you see here is a typical intraperitoneal glucose tolerance test, which you give a bolus of glucose intraperitoneally, and then you monitor plasma levels of glucose. To a little bit of a surprise, but I can explain why this phenotype. It didn't matter a lot if you have taste receptors or no taste receptors. These are whole body knockout mice, by the way, if you administer glucose intraperitoneally. But if we change the route of administration, and now we give an intragastric gavage, keep in mind with intragastric gavage, we include the intestine, but we omit the oral cavity, which of course it was beneficial to exclude potential effects that they emerge from taste. You see that glucose excursions in the knockout mice are blunted. And we immediately reasoned that this may play a role in the way by which glucose is transported or is absorbed, but there are so many ways by which one can modulate plasma level of glucose. So we performed the same experiment using stable isotopes to ensure that the glucose that we see in the plasma is indeed glucose that comes from the gut. And as you can see here, we recapitulated the effects of the experiment that I showed you before. Then the question is, what is the mechanism? Of course, Bob had already published this work implicating the role of taste receptors in enteroendocrine secretion. You see here blunted GLP-1 in Begestusin knockout mice. We show the same thing for the T1R2 knockout, but we had like a physiological problem here. That wasn't consistent. The role of GLP in this mechanism wasn't consistent with physiology, because one would expect that reduced levels of GLP will increase gastric emptying, reduce insulin secretion, increase glucagon secretion, all of which will raise plasma level of glucose, but we see the opposite phenotype. But as Dr. Clevels mentioned earlier, L cells also co-secrete GLP-2. And here you can see in response to a glucose load, this is GLP-2 in the portal vein. This is very critical because this is where you find most of the concentration of GLP-2. Slightly raises in the wild-type mice, but is blunted in the knockout mice. And that made a little bit more sense because GLP-2 is known to be involved in nutrient absorption. So we follow a mechanism around GLP-2. And I'll summarize here. So in response to a meal that is very high in glucose, that's actually a requirement, stimulation of taste receptors in the luminal side, co-secretes GLP-1 and GLP-2, and GLP-2 through neuronal stimulation induces the translocation of GLUT2 from the basolateral membrane to the apical side to pick up the extra glucose and bring it into the circulation. And we primarily demonstrated this using the Using chamber, not only in vivo physiology, but the Using chamber, which allows to measure nutrient transport in intact intestine. So one side represents the apical side, and the other side, the basolateral side, and you can add different nutrients or molecules in one side and monitor their appearance in the other side. For that purpose, we use 3-OMG as a non-metabolizable glucose analog. And you can see here, this is glucose transport in wild-type mice, but in the knockout mice is blunted. And we were also fortunate to collaborate with a number of gastroenterologists back then, where we were getting straight from the OR intact explants, intestinal explants from patients, and you can clearly see when we inhibit with lactosol, we have a general trend for the reduced 3-OMG absorption in human intestinal explants. And to further demonstrate the role of GLP-2, we gave mice, and in this case is intestinal preparations. We give teduglutide, which is a GLP-2 analog. It doesn't make a big difference in glucose transport in wild-type mice, but it rescues the phenotype of the knockout mice. And of course, as one would expect, when we use intestines from GLP-2 receptor knockout mice, we recapitulate the phenotype of the knockout mice. Okay, so pretty much what I show you, it's like a little bit expected because, you have the taste receptors, the ligands, in this case, maybe sugar, you induce an acute signaling, you have a certain response. And it seems that, so far the cellular context is what makes the difference because taste receptors in the oral cavity, they will sense sugars to release ATP. Similar cells, L cells in the intestine, they will sense intestinal sugars in the lumen to release GLP-1, GLP-2, as others have shown. And the same thing, sugars in the blood. Of course, glucose is the major sugar in the blood, will be sensed by taste receptors to release insulin. So you have the same machinery, but different contexts. So immediately, several years ago, we asked, does the function of the sweet taste receptors extend beyond the endocrine tissues? And the answer is certainly yes. Through the reporter mouse that we developed, you can see here expression of T1R2 in skeletal muscle. And that was very interesting for us because, skeletal muscle is not the typical endocrine organ. Nevertheless, it plays a very important role in metabolism and especially glucose sensing. And to make the long story short, we identify the signaling mechanism, which involves activation. In this case, has nothing to do with calcium. It's activation of ERK2, which is phosphorylation, translocates to the nucleus. It binds and phosphorylates directly. PARP1 is poly-ADP ribosylase, is an enzyme that uses NAD as a substrate to post-translation and modify proteins. That plays a very important role in DNA damage, but also it has been demonstrated plays a role in metabolism. And that pretty much governs NAD levels. NAD, of course, is a very important molecule for cell homeostasis and metabolism. And as you can see here, this work was done in muscle-specific knockout mice. Muscles respond to sucralose, but wild-type muscles, but not knockout, as you can see here. And generally, pyrylation, which is the enzyme activity of PARP, is reduced, and that leads to elevated levels of NAD. Okay, so what are the consequences of that? We block taste receptors, we block PARP, and that increases NAD levels. Now, NAD is also the substrate for another class of enzymes, sirtuins. They are very, very popular, of course, in aging and metabolism. And we show that the blocking PARP allows increased levels of NAD and therefore activation of sirtuin and downstream effects through PGC1 alpha. And that leads to the anticipated anticipated physiological consequences. I show some here. Increased mitochondria content. This is through EM. You can see more larger, robust mitochondria network, but also increases mitochondria function. This is in isolated fibers from wild type and knockout mice measuring mitochondria function. And overall, the mouse has better muscle fitness, and that has physiological consequences. These mice are better runners. As you can see here, they have higher endurance. Okay, so over the years, since I was a postdoc, it became apparent to me to understand whether everything that we study has a physiological relevance, has a translational relevance. And for that, we resorted to the pool of different SNPs that can be found, particularly in T1R2. And through work that we did biochemically and physiologically, we identified that the ILE2-191 valin SNP is a partial loss of function SNP that also has relevance to human physiology. And we use a number of cross-sectional studies to establish the translational role. But recently, we had the opportunity to perform a retrospective analysis of an older obese population that it was subjected to a 6 months exercise and weight loss program. And the reason we use this population, stratify them according to the SNP, is because we had available a number of outcomes that they were relevant to the latest work that I show you in the skeletal muscle. So as a summary here, you see these are normalized effects of the exercise intervention in a number of variables, starting from muscle mass to oxygen consumption to mitochondria function. And you can see here on the brown, valin carriers, which is the partial loss of function equivalent, they perform better in all of these parameters. And then when we perform a composite index that instead of cherry picking which variables we're going to assess, we just sum all the normalized data and we classify between responders and non-responders. You can clearly see here that every single valin participant, partial loss of function of T1R2 participant, performed better than any of the ILE participants. And we're very excited about that. At the beginning, we thought it's a statistical fluke, but we resorted to bioinformaticians that they confirmed that the chances that this is random is very low. And recently, we tapped into larger databases, such as the UK Biobank, to confirm these effects. So I'll stop here and thank everybody for the invitation. This latest work was done by Joan Serrano, and I want to bring his name. Bob will not remember, but a few years ago, 7, 8 years ago, Joan contacted Bob for a postdoc, but Bob didn't have an available position, and he sent me an email and says, "Hey, I have this postdoc. Are you interested in him?" And I said, sure. So Joan came into my lab, and in over 6 years, he published 6 first author papers, several 2-digit impact factor publications. So thank you, Bob. That shows your generosity and your collegiality. And again, thank you for your attendance.
We have time for questions. Your data on the muscle physiology is really fascinating. It seems really important also. So would the model be that during exercise, that blood glucose levels would change, and this is the response to changing blood glucose levels?
Yeah, just to make this clear. So the taste receptors in the muscle have a suppressive effect on NAD levels. So think about it. In the absence of taste receptors, in reality, you're telling the cell that there is no energy, there's no glucose. And therefore, the cell, from that perspective, does what the cell will do also during fasting, raises the levels of NAD. But instead of doing it through its NAD biosynthesis, it's doing it by suppressing its consumption. So what you observe there is chronic adaptations in response to having either partial loss of function in humans or total loss of function, of course, in mice. So it's like a break of NAD regulation, if you wish. And the beneficial effects is a side effect in essence, because higher levels of NAD will have, over a long period of time, the anticipated beneficial effects in making the muscle better, right? Through mitochondrial function, through endurance and metabolism.
Well, that just makes me wonder if there might be a role in cardiac muscle as well. I wonder if you look there.
Yes, we have looked there, and especially that's very relevant, not only because of NAD, but also because of the role of part 1 that plays in.
PGC.
Yes, absolutely, PGC1 alpha. We have manipulated mice, and we haven't found yet some robust phenotype. But I don't think we have done all the typical TAC experiments that they may be required to stress the system and then manifest. We have done half a diet. We see some difference in hypertrophy, heart hypertrophy in OB models, but nothing robust. But the expression is there, and it's actually quite selective. It's not in all fibers, in cardiac fibers.